IIT JEE विद बबीता पर आज हम करने वाले हैं सेल द यूनिट ऑफ लाइफ का पार्ट 3। पहले पार्ट में हमने सेकेंड पार्ट में हमने प्रोकैरियोटिक सेल्स कंप्लीट कर लिए थे। नाउ वी हैव डिफरेंसेस बिटवीन द प्रोकैरियोट्स एंड द यूकैरियोट्स ऑन द बेसिस ऑफ डिफरेंट पैरामीटर्स। So the first of the difference we are having is on the basis of cell wall. So in prokaryotes, cell wall is made up of peptidoglycan, except for mycoplasma, where the cell wall is absent. So in mycoplasma, no cell wall is there. Then it is non-cellulosic. Cellulose की नहीं बनी होती है. It is just made up of peptidoglycan strands. Then in eukaryotic we have different materials to be used for different kinds of cell walls. So first of all we have protists. In some of the protistas, cell wall is present with different compositions. So in some of them, cell wall is present. In fungi it is present where it is called as chitin. It is composed of chitin, and chitin is called as fungal cellulose. It is due to the special property of chitin to comprise the cell wall of fungi, because in plant cells cellulose is the composition of cell wall. So in fungi we call it as chitin is fungal cellulose. Then we have plants. In all plant cells, cell wall is present and made up of cellulose. Then in animals, for animals, a cell wall is absent. So the outermost layer in the animal cells is just the plasma membrane, which is universal in all kinds of cells, whereas in an animal cells, cell wall is absent. The second difference we have is on the basis of surface appendages. So there are 3 surface appendages as we read in prokaryotes. First of them is pili. Secondly, we have fimbriae. And thirdly, it is flagella, the longest strand of all which we saw there. Then in pili, in prokaryotes is present, in eukaryotes is absent. In fimbriae is present in prokaryotes but absent in eukaryotes, just same as pili. Then we have flagella. So here it is made up of flagellin protein. It is present in eukaryotes as well, but here it is made up of tubulin protein. Flagella is a surface appendage that is present in both prokaryotes as well as eukaryotes, but their composition is different. In prokaryotes it is made up of flagellin protein, and in eukaryotes it is made up of tubulin protein. Another difference on the basis of flagella, so here the flagella has 9+0 arrangement, and here it has 9+2 arrangement. क्या होता है आगे पढ़ेंगे. Next we have difference on the basis of mesosomes. Now what are mesosomes is that the little structures we observed in the structure of prokaryotes were mesosomes. So those are present in prokaryotic cells but absent in eukaryotic cells. Now we are going to differentiate on the basis of ribosomes. So prokaryotes have 70 S ribosomes, which are not at all additive. These are made up of 2 units, 30S and 50S. The sum of 30 and 50 becomes 80, but these are not additive, so it becomes 70S here. Then it is present in plasma membrane and cytoplasm. So it is present in cytoplasm and plasma membrane. Now in eukaryotes, ribosomes are ATS type. ATS type in cytoplasm. Again, ATS is made up of 60S and 40S, which is not at all additive. And 70S ribosomes are also present in mitochondria and chloroplast, which is only present in plant cells. So mitochondria is present in animal cells, plant cells, protists, and fungi. Whereas chloroplast is present only in plant cells. The next difference we have is on the basis of vacuoles. So in bacteria, ट्रू और सैप वैक्यूल्स जो अभी लास्ट टॉपिक में पढ़े थे. For reference, refer to part 2. So true vacuoles or sap vacuoles are absent in eukaryotes, whereas gas vacuoles, prokaryotes, whereas gas vacuoles are present. So gas vacuoles are present. Here in eukaryotic cells, true vacuoles or sap vacuoles, as we call them, are present. So here we have presence of true vacuoles or sap vacuoles. So the next reference we have is on the basis of nucleolus. So nucleolus are called as ribosomal factories because ribosome is synthesized in nucleolus only. So these are known as ribosomal factories. Nucleolus are present in more in number and bigger in size in cells which are actively involved in protein synthesis. Then these are absent in prokaryotes but present in eukaryotes. The next difference we have is on the basis of DNA. So in prokaryotes we have double-stranded circular DNA. Here DS stands for double-stranded circular DNA. So it is coiled with the help of polyamides. Coiled with polyamides. Polyamides are non-histonic basic proteins. Non-histone basic proteins. These are polyamides. Then we have circular DNA, which is called as genophore. Genophore. It is also called as prochromosome, as we studied in the topic of prokaryotes. The DNA in eukaryotes is also double-stranded, but it is linear DNA, coiled with the help of histone protein. Coiled with histone proteins. Coiled with histone protein. Just as polyamide, it also has a basic nature, because to hold down DNA stands for deoxyribonucleic acid, and to hold together acids we need some basic substance. Then we have another difference on the basis of RNA. So we have the difference on the basis of RNA. So RNA is single-stranded. There is no difference between difference in RNA in prokaryotes and eukaryotes. So here is single-stranded. Single-stranded. And again here we have it as single-stranded. So this was all about the differences between prokaryotes and eukaryotes. Now let's head on to the next topic. Now we have eukaryotic cell. So in eukaryotic cell, the two major parts we have is plant cell and animal cell. So the major difference between plant cells and animal cells is on the basis of size. So plant cells are larger in size than animal cells. So animal cells are smaller in size than plant cells. Now we have difference on the basis of cell wall. So in plant cells, cell wall is present, cellulosic in nature. In animal cells, cell wall is absent. An outer covering is plasma membrane. Plasma membrane is the outermost covering. The next difference we have is on the basis of plastids. So plastids are present in plant cell while absent in animal cell. The next difference we have is on the basis of vacuoles. So vacuoles are larger, central, and permanent. Sap vacuoles and one in number in plant cells. In animal cells we have many small temporary vacuoles such as contractile vacuole in amoeba, contractile which is meant for excretion in amoeba, and food vacuoles. These are some vacuoles present in animal cells. These are spread all over the plant cell and are many in number. In detail, we'll study in the concept of vacuoles in eukaryotic cells. The next difference we have is on the basis of nucleus. So in plant cells, we have the nucleus at periphery. There's a logical reason behind it. Suppose this is a plant cell. This is maybe this is its cell wall and this is its plasma membrane, just assume. We'll do it in detail. So the middle portion of it is covered by vacuole. So the nucleus gets a small space at the periphery. This is the nucleus, it gets a small space at the periphery. That's why the nucleus in plant cell is on periphery. In animal cells we have whole of the space empty except for some organelles spread over in the cytoplasm. So nucleus gets a respective seat in the center. So we have a central nucleus in animal cells. Now we have differences on the basis of Golgi apparatus. So in plant cells, Golgi bodies are simple and are called as trichosomes. The cisternae in plant Golgi bodies are quite away from each other. So these are away. Whereas in animal cells, there is a single and complex unit. Of the Golgi bodies. So here it is. The cisternae are near to each other. Here we have this way. It's quite complex like this. The cisternae are somewhat rounded, but for easy explanation to show you nearby, I just did make them this way. Next we have centrioles. So centrioles play a functional role in division of cells. So they help in cell division. Now do ask me why, how. So they radiate spindle fibers. You'll read in the chapter cell division in cell cycle. So they help in radiating spindle fibers. So these are absent in plant cells and present in animal cells. So they have a formation like this. This is an animal cell, just assume. Here we have chromatids. क्या होते हैं फिलहाल के लिए नहीं जानना. As I told you, you'll read them in next chapter, that is cell cycle and cell division. So these are centrioles. These radiate spindle fibers this way. The spindle fibers attach to the chromosomes. They pull them apart. So the chromosomes pull the chromosomes apart. This way centrioles help in cell division in animal cells, whereas they are absent in plant cells. The last but not the least, we have difference on the basis of shape. So plant cells have a fixed polygonal shape. The shape is fixed because they have cell wall present in them around their cells. Whereas in animal cells there is no cell wall present outside the cells, so they have an irregular shape. So friends, the outermost structure present in all eukaryotes universally is plasma membrane. It had been in prokaryotes as well, but there it had been as third structure. After glycocalyx, then bacteria, and then plasma membrane. Here it is in animals we find it is the outermost layer, whereas in plants it is the second outermost layer after cell wall. So it is living in nature and universal. Universal, how it is, I've discussed with you. Then it is thin, delicate. Thin means it is very low, very less in amount. It is delicate, so soft and elastic. Elastic इसलिए because when the cell grows, when the cell keeps on growing, the plasma membrane needs to stretch as well to keep on with the balance of the growing cell, with the boundary of the growing cell. So it needs to be elastic, and it has to be, so it is. Then it is selectively or differentially permeable in nature. Including me, I found several students confused between semi-permeable and selectively permeable. So I found a non-confusing answer for you between selectively permeable and semi-permeable. So selectively permeable is the one which allows entry or exit of selected items, means all solvents and some solutes. The solutes are selected, so it is called as selectively permeable. Where semi-permeable allows only solvent, means a solution is composed of two parts. One is solvent and the other is solute. So the semi-permeable allows only half of that, as the word semi represents half. So it allows only solvent to pass through itself. So that is the difference between semi-permeable and selectively permeable. For neat purpose, mere words matter. So here selectively and semi has a great importance, especially for statement-based questions. So here the most important thing in plasma membrane to remember is the difference between selectively permeable and semi-permeable. Then its composition, so it is made up of two things, made up of lipids and proteins. So made up of lipids and proteins. Lipids are arranged in a bilayer with hydrophilic heads outside and hydrophobic tails inside. That too we are going to learn very soon. And proteins are spread throughout, intrinsically and extrinsically throughout the plasma membrane in a non-symmetric manner. And it is universal. It is the absolute requirement or the basic necessity or prerequisite in scientific words. It is prerequisite for all living organisms, including the prokaryotes, eukaryotes, plant cells, animal cells. The last but not the least, it deals with the outer world. Even if in plant cells the cell wall is the outermost layer, but cell wall allows most of the items inside except for some cells, for some objects which are bigger in size. So otherwise it allows everything inside. Plasma membrane has to select which thing has to go and which thing has not to go, inside and outside as well. So it can be called as the gateway of the cell. Gateway of the cell. Because it is the major decisive for whatever has to come inside and whatever has to go out. What we have now are different models of plasma membrane, which represent the structure of plasma membrane. So they represent the structure of plasma membrane. Hope you understand the abbreviations I use. If you don't understand, refer to the words spoken by me. So there are different structures of plasma membrane given at different times. So the first of all was trilamellar or sandwich model. Now you would think, how did it be called sandwich model? I hope everybody of you has eaten sandwich once at least in your life. So what sandwich comprises is one layer of bread, then another layer of stuffings, and again a layer of bread. Then this represents trilamellar. Here refers to layers. So trilamellar refers to 3 layers. Sandwich has 3 layers as well, as I told you right now. So it was given by Danelli and Jefferson. Danelli and Dawson proposed this model. It states that plasma membrane is a 3-layered structure. 3-layered structure. Hence was it given the name of trilamellar model. It says that it is composed of lipids and proteins. As we discussed in features of plasma membrane here, lipids and proteins. So it also says the same thing, that trilamellar layer says that plasma membrane is made up of lipids and proteins. It has 3 distinct layers, out of which 2 layers of lipids, which represent the bread of the sandwich, bread, and a layer of proteins, which was wrong, as I told you that proteins are spread throughout, so it was wrong. But they said that it is the layer of proteins, it has a layer of proteins, which represents the stuffings. So just because the proteins are not spread, do not represent a layer and are spread throughout, so the model was rejected. So it was rejected. So what we have is here is the diagram of the sandwich model. So these are two outer layers of proteins and this is the inner bilayer of lipids. It is also called as PLLP model, where P represents the first layer of protein, last P represents the third layer of protein, and LL represents the inner bilayer of lipids. Then it had certain drawbacks as well. Had it not had drawbacks, why would have it been rejected? So it has certain drawbacks as well. So first of all, it states that the membrane is rigid or static. Membrane is rigid or static, whereas in reality it was wrong. The membrane is elastic. It has to cope up with the growing size of the cell with time. The next we have is, it said that the membrane is symmetric, whereas the proteins are spread throughout in a really asymmetric manner. So the membrane is asymmetric. Due to these two statements, the theory became wrong. So fluid mosaic model was the second model of plasma membrane. It was proposed by Singer and Nicholson. Remember, Singer and Nicholson in year 1972. Usually in biology we don't pay attention to the years, but here we'll have to pay attention to this 1972, just because it has been previous year questions so many times in it, and it is the most widely accepted model. So most widely accepted model of plasma membrane. It says that chemical composition of plasma membrane is not fixed. It says that plasma membrane composition varies in different cell types, varies according to the variation in cell types. Here I have some examples for the composition of plasma membrane in different cell types. First of all, we have lipids, then we have proteins, then we have carbohydrates, then we have enzymes, and at last we have water. So we have enzymes and water. So the composition of lipids in fluid mosaic, according to the fluid mosaic model in plasma membrane, is around 20 to 79%. Varies according to the variation in cells. Now we have protein, so 20 to 70%. Again varies according to the variation in cell. Then carbohydrates vary from 1% to 5%. Enzymes are about 30 different types. There are about 30 different types of enzymes which compose the plasma membrane. Then water is around 20%. Then we have a plus point which is quite important. So in human erythrocytes or human RBCs, plasma membrane in human RBCs or erythrocytes, as we call it scientifically, in those the plasma membrane is composed of two different types of substances. First is proteins, and the second one is lipid. So we have proteins and lipids. So proteins are 52% and lipids are 40%. Rest 8% are composed by these carbohydrates, enzymes and water. So to remember it is an easy way. 52% proteins, 52 is the larger number and protein is the larger name. Whereas 40 is a smaller number and lipid has a smaller name. So pay attention to this 52 and 40 to remember proteins and lipids. I've told you in easy way. So lipids in plasma membrane are basically three types, varying on the basis of composition. So first of all we have phospholipids. In these phospholipids, the lipids perform two major functions. First, they form backbone of plasma membrane. So they form the backbone of plasma membrane. Secondly, they are arranged in bilayer. These are the two major functions of phospholipids. Means one is function, second is characteristic. Then these phospholipids or phosphoglycerides, as we refer them to as. So phosphoglycerides. Here we have cephalin and lecithin. Cephalin and lecithin